Good morning, students. Welcome back to lecture 10. Today we will talk about a little bit more on HLA typing and tissue transplantation. In the last class, we introduced the results between structure and function of MHC molecules, HLA, why it's called as human leukocyte antigens, and a little bit more details on HLA polymorphisms, types of HLAs, types of gene segments, different polymorphisms in terms of haplotypes we looked at. Again, little bit more, I will recap that concept again to better understand the organ transplantation. So this is overall summary of the unit 2 remaining portion. We discussed last class HLAs and polymorphisms. We'll recap again. Then we will bring two concepts specifically: tissue or organ transplantation, tissue typing methods for tissue and organ transplantation. How do you identify a match in the tissue? What are the important basic concepts in terms of how do you choose a specific organ from a donor to recipient? What are in terms of physiological, immunological match conditions between the organ and the recipient, donor and recipients, and techniques in terms of HLA typing, serological testing, and some other molecular based tests are available now. We will look into that and then we will conclude. On Saturday, we will look into a little bit more on videos in general. Then the remaining class in terms of probably on Monday, we will take on concepts of graft and host rejection, GVHD or graft versus host diseases, and xenotransplantation and immunosuppressive therapy. We may take this as a together one class, or given the time, we take only graft versus host reaction one class, further followed by immunosuppressive therapy. Better to go for two classes. Don't need to rush. Probably next week we will cover unit 3. Okay. Again, basics we already discussed in last class. We will look into further more slowly. Histocompatibility antigens or histocompatibility complex, specifically they are called as antigens, known as transplantation antigens. They were first identified in transplantation experiments in mice. It was done around 1900s early. And at that time, in terms of antigen and antibody, what is the protein, in terms of the molecular or biochemical characterization of antigens and antibodies was nothing available. Only they know that from the anti-sera, you can raise some antibodies, or you can do some grafting experiments, remove one tissue from one mice to the other, whether it accepts the tissue or not. If it doesn't accept, there will be a rejection mechanism. They were basically studying those concepts at the time without understanding the biochemical and molecular nature of antigens and antibodies, physically based on the phenotype, accept or reject. So that's why at that time only they found some molecules which are important role for tissue compatibility, because some specific group of genes that are responsible in the host or in the recipient from the donor, they are called major histocompatibility complex genes, because if the genes are acting properly, or they have the capacity to recognize the foreign tissue attached to this mice as self or non-self. If it is recognized as non-self, it becomes histoincompatible. There will be a rejection or some reaction happens on the mice. And then if it is compatible, it matches with the recipient. The mice looks normal and healthy. Based on this concept of compatibility, histocompatible, these genes are identified. And there is one more minor class is there, but one more important class they identified was major histocompatibility complex that helps in transplantation. They are called antigens because we will see in further slides why they are called antigens, because they are helping T cells in cell-mediated immunity that we already looked up. So they are called HLAs in terms of coming to human side because they are specific to humans, major histocompatibility in terms of major, and they are mainly proteins present on the leukocytes, white blood cells. That's why they are called as human lymphocyte antigens. They are antigen because they are self molecules present in the body. So we looked at again a little bit more on HLA polymorphism and gene structure, location in chromosomes. We will recap again the concepts. What are HLA genes and what molecules? The MHC are synonymous HLA is characterized by its polygeny present. That means the different genes encode different MHC molecules, and they are highly polymorphic based on the concept of more genes present. At the same time, the concept of haplotypes or codominance, we will see that what is codominance. And the loci coding class 1 and class 2 molecule display the highest level of variation, that polymorphism. We already looked in the classes, class 1, class 2, and class 3. Class 1, we looked at little bit more detail. For example, one of the class A MHC HLA-A, it codes for 566 or 800 polymorphic genes, haplotypes that we looked in the last class. That's saying that MHCs are a cluster of genes which is generally inherited as a complete set called as haplotypes from each parent. So why we are telling each parent? Because the concept of codominant expression comes up. We get different chromosomes or different set of chromosomes from paternal and maternal. So we are just simply saying why the codominant saying is that if given a haplotype of HLA gene, it coming from both mother and father, and different sets of haplotypes based on the paternal, maternal will be there in the chromosome, and they are equally expressed. It's not saying that father's haplotypes are more dominant than mother's haplotypes. They work in a codominant fashion. Together, they express certain set of genes, that's haplotype responsible for that. They regulate MHC regulation in terms of gene expression. What is codominant expression? That clearly MHC genes are expressed in codominant fashion, i.e. alleles inherited from parents, one from father, one from mother, are simultaneously and equally expressed. So if allele from father, mother, they are not same, they are different. That's why we are calling haplotype. And it doesn't mean that the difference will give a favoritism, that father is coming from father should be more dominant in terms of expression, make the mothers recessive. It's not like that. They're simultaneously and equally expressed, making a regulation of MHCs completely controlled by genetics. We look into in terms of details, again more abbreviation we looked, class 1, class 2 MHCs, we looked at that, what is the function, specifically helping the T cells. You again go into brush up, class 1, class 2, class 3, they are different location. Order is different on the chromosome. If you take each class, there are different genes, that's polygenic segments. And you look into more in the HLA protein structure, we already seen that class 1 will have different alpha units and only one beta helix. Class 2 will have equally. And then we spoke about this peptide binding groove that is really important, helping the MHC to process and present the antigen to T helper cells. This is the peptide binding groove, very highly polymorphic and highly important for the function of HLAs. That we already looked. In terms of here, only alpha helix helps. In front of this class, both alpha and beta helix will help in peptide binding. And coming on to polymorphism, we are suggested, we already looked in different polymorphism, A, B, and C, DR, DQ in the class 2. And then you looked at that A and B, what are polymorphism present. This is of 2014 data, but now with SNPs and whole genome assembly, we are getting a lot of haplotypes. A lot of alleles have been added every time. For example, every day some 200 alleles have been added. So the research is going in a very progressive way. That coming on to functions of MHC molecule, the primary immunological function is to present the antigen peptides. They will bind to the antigen peptides and they will present the antigen peptides to specifically T cell receptor. And how they will do CD8, CD4, class 1, class 2, we already discussed in previous classes. And you look further into that, one more important concept of antigen processing and presentation. So we looked in previous classes that one coming from the pathogen, you break the pathogen, you go through lysosomes, mainly one the MHC2 will bind that is called as endocytic pathway for exogenous antigens. And then endogenous antigens already in the blood that consists of cytosolic pathway that directly absorbed here. But you need to break here. Fundamental difference, you look into that. And in terms of processing, the process of the antigen, you refer to degradation of proteins into small peptides, fundamentals of biochemistry. Presenting refers presenting it in a form where T helper cells possibly recognize that. So basically, we suggested there are multiple tons of antigens from microbes. So we already know that the polymorphism and polygeny helps to make specific MHC attached to that. Based on that, specific T cell will come up. How they will do that? We looked into a specific complex, that ternary complex should be formed. This looking into a T helper cell. This is the antigen presenting cell, for example, dendritic cell that has a class 2 MHC molecule. And you know that 2, 4, 2 is 8. 2 will always associated with CD4. And this is the TCR. And this is the peptide already processed bound to the class 2 MHC molecule that is represented to the TCR. This ternary complex formation is important. At the same time, how this processing in terms of processing of the peptide. That's why there are specific genes called they help in processing and presentation of the antigen. In class 2 region, they are called LNP, TAP. LNP encodes for proteosomes, TAP encodes for transport peptides from cytosol to ER, and tapacin encoded in peptide loading. So that's why there are specific genes, proteins helping to compensate the function of MHC molecule. So this is an important slide. You look back into in terms of your 2 marks, 4 mark question in terms of difference between MHC1 and MHC2 molecule. You look into the criteria here that we already discussed several times. You go into specific MHC and MHC2 molecule, write down the notes, cytosolic endocytic pathway. An important thing, CD8, CD4, mnemonic we used. One more common understanding that these MHC class 1 are present in all nucleated cells in the body. That's why it's called as ubiquitous expression. They are everywhere. Every cell in the body will have it, except sperm cells and platelets. Except sperm cells, leaving that all nucleated cells and platelet cells will have MHC1. Coming on to class 2, they are mainly present on antigen presenting cell. What are they called? APCs. Dendritic cell, macrophages, few other cells we discussed. They are APCs. They are restricted here. That's why they are inducible expression. These are constitutively expressed, represent all over the cell. One more thing why we are saying that this ternary complex is important, because it provides a specificity in terms of you are looking for the type of HLA here and the same HLA here, but from here to here the antigen has been changed. But once a T cell binds to this, makes a memory of it, it cannot bind to another antigen. Though the HLA is same, MHC is same, it's a different antigen, but it cannot bind back. But if you make a different HLA and same antigen also, T cell will not bind. This highly specific, this concept of MHC restriction is important. That's why polymorphism is important in terms of specificity. Okay, that finishes our recap a little bit more. Hopefully you got it understand better compared to last class. Now why we are talking about this? Because we are talking about fundamental concepts of MHC making self, non-self recognition in terms of the body. That is really fundamental for organ transplantation or tissue transplantation. That will be the interest for further class now. This is one of the noble deeds humanity as such we are doing. The organ donation is one of the noble deed can be considered, helping the person who is under a disease condition, who is in need of a healthy tissue from a healthy individual or donor, or from a cadaver. We will see that it can be done from healthy or cadaver, and that high community it's combination of in terms of scientists, in terms of immunological studies done, in terms of model organisms also, in terms of mice, how it helped to understand the grafting processes. It is a culmination of a human effort. And here in an example, you are seeing in a person who is a recipient, you received a kidney transplant. The disease may be a kind of self from the body. This may be the foreign. They can communicate with each other in harmony. That means saying that, hey, I am accepting you. We are go ahead. We can go ahead with normal functioning. I don't want to kill you through my MHC or T cell activation. So there is a tissue compatibility happens here. How does it happen? We will see that. An example may be seen that the HLAs responsible for this kidney and other haplotypes, they should completely match with the HLA coming from the recipient. If the match is absolutely nice, with minor issues is fine, but it matches the more better, the more good the transplantation happens. If it doesn't match for say completely different HLA type from here to here, basically they will be considered as, based on the polymorphism, they will be considered as foreign and they will be eventually killed. So you need to have a HLA matching, that we will see in further. How do you do HLA typing? Fundamentals of organ transplantation. Organ transplantation is the transfer of healthy organ or part of it from a donor to a recipient, and at the same time while re-establishing vascularity. Because some of these organs transplanted, they may influence the hematopoiesis, they may influence the blood production in the recipient. So the initial change may be temporary. It may again adjust to the condition of the recipient in physiology and immunology, but it takes some time to re-establish the vascularity in terms of normal blood cell function. So organ transplantation is transfer of healthy organ or part of it from a donor to recipient while re-establishing vascularity. The first successful transplant, it was done in kidney in 1994. Further, there is significantly increased success rate because of improvement in surgical techniques, improvement in immunosuppression technologies in terms of HLA matching and all. We will see in the class. Several organs are transplanted, but lack of organs, the prerequisite, makes it difficult to do the job properly. What are the organs available for transplant? There are several organs in the body. You can use part of it. Kidney, liver, lung, heart, pancreas, intestine, and coming of the tissues. Skin can be transplanted. It can be grafted also. And then the fundamental process, how a transplantation works. The cell is happy. It has a self-MHC marker, MHC in terms of self-marker MHC, that is a haplotype. That is known polymorphic, that is self-polymorphism. It knows it is producing those HLA polymorphism. It knows that all the variants it knows, and it says, I'm happy with it, I have no problem. But either the antigen or either a MHC molecule that is polymorphic to this, for example, HLA A class, but this will have other polymorphism. It may be a kind of numbers D and 01. This may be number D and 04. We will see the nomenclature. Then the cell is unhappy, it takes it as a foreign, it kills it. But if that is the case, the tissue or the donor is not fit for transplantation, you will keep searching for where the marker or MHC polymorphism matches with the other person in terms of the donor, and there is a healthy transplantation happens. This is the fundamental process of transplantation. And as said, it's not an easy job to take some foreign thing and make it as your own. Though we are humans, though we have the match, but there is some physiological, immunological consequences that have a lot of, in terms of, for example, the donor may die if you are giving a like incompatible tissue or incompatible immune cells. So there are a lot of terms that you can use for organ transplantation. Fundamentally, whether organs are available for transplant or not, this is the fundamental prerequisite for any transplantation. Then you look for recipient and what kind of transplantation here is required. Then you look for donor and recipient compatibility, you look in that. And then you also look for what are the transplantation barriers between this donor and recipient. If the match is there also, what I can think of, what will the things that go bad? You look into the transplantation barriers, but understanding fundamentals of immunology. There is a concept immunosuppression that we will cover in next classes. Further complications, the major complication may be death. The minor complication, you can use some drugs to control that. But important thing, donor and recipient compatibility in terms of their individual physiological, individual haplotypes. The haplotypes HLA matching should be 99.99% or 100% to make that proper. So if you take stages of the recipient of an organ transplant, for example, you go in terms of evaluation of the patient, what is his requirements. You make the requirement, you match what he wants: a kidney, he wants a heart, he wants a liver. Then you put it in a database, you wait for the organ to come up. Then you do a matching process. You work on the policies from the recipient and donor because you deal with humans, so you need to go for ethical procedures. So when it comes to cadaver, when a deceased organ donor is identified, who volunteers to donate his organs after his death, the most suitable transplant candidate on the waiting list is selected by matching against the donor characteristic. So when the cadaver comes, the organs are isolated from it. Then you go to the database. You go and you don't give it to anybody who wants. You have to give into the database who is waiting in the list, because the waiting list may be sometimes very high and the patient may be dying soon. So you need to give it to the immediate person who requires from the cadaver in a case. Each organ has its own allocation system based on precise criteria: recipient's age, health stature, organ size, blood and tissue compatibility, HLA typing. And when living donors are considered, selection is done upon donor and recipient matching and other problems in terms of the preservation of organs that you look into that. Cadaver donor organs, living donors. In terms of compatibility, you first look into blood ABO blood group compatibility. That is fundamental, so that rather than you give some organ, you kill by organ through immune activation. If you are matching not properly with the blood, then because these immune cells is going to make the blood later, they are going to graft, they are going to we will see what that process is that. So if they don't properly stitch with the vascularity, you are giving new blood cells into the recipient. Though the matching is there in the immune system, the new blood cells are formed, they are going to not match and they are going to kill. So basically you need to have ABO blood grouping, first blood group matching, then HLA typing. You go with HLA type. The HLA antigen, small variability can be tolerated depending on the organ. We will see that. But you go from first ABO grouping first, match the blood groups, then match the HLA types, then size match. Then in terms of the suitable donor, what is his health condition, whether he has a presupposed conditions of infectious diseases, then you is out of the scenario. Whatever matches do, you cannot give this to the recipient. In kidney transplantation, if any mismatch, there is a hyperacute reaction. Immediate death of the donor happens. We will see in the next class in grafting studies. And a cross-match test will be performed before you do the kidney transplantation. So fundamentally, not to kill the donor, kill the recipient. So that comes to one of the concepts that discussed in the curriculum, the tissue typing. Tissue typing is a procedure in which the tissues of a prospective donor and recipient are tested for compatibility prior to transplantation. You don't need to give the tissue immediately to the recipient, but you have to go with some other in vitro techniques. You go into the literature, you go into the procedures, and then you develop a concept of suitability, and then you go for after transplantation. So mismatched donor and recipient tissues can lead to rejection on the tissues, and if it is a severe, it leads to death. What happens in during tissue typing? Specifically in tissue typing, an individual's HLAs are identified first. Let's say a kind of recipient X and a donor Y, and they come to X. You look into the X. You need to know what are the HLA polymorphism in its own from these immune cells in terms of the class 1, class 2, and class 3. How do you do that? Then basically you need to genotype him. You need to genotype him in terms of immunology and in terms of HLA polymorphism present in the recipient. Then only, right, you can go for the match. If you don't know his HLAs, how do you randomly go? You randomly check, unless he survives or not, he may die in the first transplantation itself. You are going in a blind manner. You cannot do that. First, catalog his HLAs. Then you go into the database or search for HLA typing, HLA organ, then match it. So the HLA molecules, we know that they are adaptive immune response. They are present on the antigen dendritic cells. They help in T cell process. And if HLA from the donor is recognized by the recipient immune system as different, so if the recipient HLA polymorphism is different from the donor, then basically it is not a match. An immune response will come, and then donor tissues will be tissue will be rejected. So there should be a match in the HLA. More specifically saying that HLA mismatches between organ donors and recipients can lead to development of specific form of antibodies called anti-HLA donor-specific antibodies, DSS. So once these antibodies are formed through HLA mismatching, what will happen? Antibody-antigen interaction will happen. Basically, that will lead into classical pathway of complement system. So basically, I am recognizing them as foreign. So I go in terms of producing complements. Basically, I'm killing the donor tissue, right? So whenever in the body the DSAs are produced, it simply says that the HLA type is mismatched. So you cannot give these organs. So how do you do that? What are these testing for this? By using this antigen antibody crosstalk, you can do in vitro studies. You can do blood test. You can do HLA typing test based on random antibodies. You check randomly with the recipient's HLAs. If they match, you are testing it in the in vitro in a glass slide. You are activating complement system. You are killing some cells, whatever you are adding there. So tells that this is the antibody for this HLA type, and this HLA type is available in the database or not. You are indirectly using this antigen antibody interaction to identify HLA of the recipient. That we will see in the next slide. So DSAs are strongly associated with rejection. When you have in-body antibodies, the donor-specific antibodies, it's highly foreign. It will be a rejection, an indicator of antibody-mediated rejection, classical pathway. And when donor and recipient HLA are matched, then no antibodies will be produced. Donor tissues are likely to be a match and accept. Please try to understand this slide. If you understand it better, it will all make clear for you in terms of understanding transplantation, in terms of fundamentals of HLA typing. So what are the first methods we used? That's as suggested, the glass slide, serological typing. They used, what they have done is that in this technique, a donor's blood cell, donor Y, his blood cell is taken and are HLA typed by mixing them with specific antibodies. You are doing it in vitro. You have a pool of antibodies with you already based on general polymorphism available over in terms of haplotypes in the population. You made some in vitro antibodies for all, and you are just taken the donor blood cells. You don't know the HLA type of the donor. You are testing in the glass slide. You are adding antibody one at a time. Then you see that if the cross reaction happens, if it's a cross reaction, it's a match, then complement activation will happen. Then it's positively recognized by tryptophan blue. Then basically the cells are killed here, and this is a method of identifying donors anti-HLAs, and then based on this complement activation. Once you have a match of the donor, what is the HLA you identified through antibody here, then you match it with the recipient's HLA type. If they are absolutely matching, then you can go ahead with the transplantation. It will not elicit in vivo in the paired recipient. No complement will be activated because it will be recognized as self. So the method has been used widely, but it's a huge success in terms of the cost, simplicity, and low cost. But at the same time, there is a variability of HLA alleles, right? Given a patient, given a donor, given ethnicity, you might have to be testing these anti-HLA bodies to find the right HLA in the donor. You might be doing a 1-year study because you need to go for highly polymorphic alleles, highly variability. By meanwhile doing that, the recipient may die. He needs the transplantation in 1 week. So how do you go that? How do you use same old serological technique? So there is some advances in terms of genotyping, in terms of molecular biology. We will see if time permits in the given class today. So this is again in vitro what we've spoken. You have a kind of from the donor blood cells here. You don't know the HLA types. This blood is in vivo, but your testing is in vitro on the glass slide. You add one antibody at a time. If HLA X, this antibody is anti-HLA B27, and it's the cell from the donor blood will have it, then it will elicit a complement process. It will give a blue signal. Then based on this, you say that one of the well has HLA B27 antibodies. If they don't have it, then you don't get this another HLA B40. It's not there here. Then there is no complement. You added the antibody, no reaction is happening. So you can say that this donor doesn't have HLA B40, but he has HLA B27. This is a match. You can match it with the recipient and go ahead. So basically that's what we've spoken, complement dependent cytotoxicity. It's also called microlymphocytotoxicity. It was developed in 1960s, and few other molecular typing we will see in further slides. So this is again one more cellular level, you call it as mixed lymphocyte culture, where you use it to identify class 2 molecules of donor and recipients. Basically you are saying that class 2 are coming from the antigen presenting cells, right? They present a specific class of T cells given the donor and recipient. You take either donor or recipient, you try to find a match of T cells through MHC molecule that you look into here. This slide, you go a little bit more. It may be not necessary right now, given the time. We'll come back to this slide again. It's basically saying that the HLA types from donor and recipient matched or not matched. If they are matched in terms of antigen, HLA-A2 matched donor recipient. In terms of allele, what we are saying is that HLA-A, allele 2, 1 is complete match here. So you can use this recipient, this donor for this recipient. If any of the allele or antigen mismatched, you cannot use them. These two are rejected. So coming on to what we are speaking here, this is nothing but a specific HLAs can be given a nomenclature. They have a specific, what do you call, just like binomial nomenclature, because there are polymorphisms. There are many of HLAs are available. You need to have a catalog for that. What is there used in terms of, you go into this nomenclature, but I will go into at the end of the slide, what is the designation? You read a little bit more time on this first and second. It's just a descriptive manner. So just these first two, you cut off here. This is the general. You're talking about HLA here. That's, and then HLA is human leukocyte antigens, a class of genes, your molecules you're speaking. And then this is the locus. Specifically, you're talking about class 1. You're talking about A here. And then you're talking within the A, another polymorphism. There are multiple polymorphism. You're talking with the number 24. It corresponds to serological antigen. You have done by blood testing, serological testing. You know that this is a 24 antibody I added. Then again, the antibodies may have subtypes based on the alpha and beta. It may be a class called 02. These are different locations for other substitution, but forget about that right now. You look into HLA-A 24-2. This is one of the antigen based on A locus, type, subtype. HLA typing is coming on to a little bit recap. HLA typing in this test, donor antigens expressed on the surface of leukocytes or their genes are matched with that of recipients. Donor antigens, what are the HLAs present in that, MHCs, they should absolutely match with the recipient, then only transplantation is successful. The closer the HLA antigens, the organ is not rejected and you can go ahead with that. Then how the matching will happen, they will look into that. Usually, whether you will go for one HLA match or two HLA match, how do you do that? There are so many polymorphism. How can you define class 1, class 2, class 3 matched or not? Given a time, you cannot go with testing all the polymorphism, but generally immunologists have they suggested that you can either go with 6 of these HLA antigen polymorphism, test that. If it's kidney, you go with the 10 testing for HLA antigen within donor and recipient. 6 is a good number. You go with the compatibility. And coming on to siblings, you may go with 25% match as suggested by here. One of the concept here looking for here, mother HLA haplotype, father HLA haplotype. You see into that, the mother is again diploid, chromosome 1, chromosome coming from mother's father and mother. You see that there is a variability in terms of haplotypes of HLAs. It's 942 here, in another chromosome 3615. But father, they will have different repertoire. But again, we go with fertilization, recombination. It's mistake here. It should be taken mother, this chromosome. It's actually a child. Mother takes this chromosome in recombination, 942. Father takes this chromosome, that's 1719. This is your haplotype, for example, coming from your parents per se. It differs from that. You get a match of 50%, but within the siblings, you get a match of 25%. That's if you're looking for a donor from within the family, you go into this kind of haplotypes, and you don't need basically any tests for these to do. Basically, if you are a sibling, 25% will match by default. And if you are a mother, 50% will match by default. But whether they will in terms of immunologically compatible or not, because there is complications, you test it through in vitro and other tests, you can accept that, yes, you can take the kidney from the mother per se here. So this may be again reading slide, you go into that. This is haplotype matched with the same locus, same loci, same order, A3, B7, DR2. But if the order slightly mismatches, for example, reversal of any of these locations, it's again saying that it's a different haplotype. This location also important for recombination. They may not take the match. HLA matching is really important. HLA matching promotes the growth and development of new healthy blood cells that are called engraftment, we will see, and it reduces the risk of post-transplantation after that's one more process called graft versus host diseases that we will see in the next classes. In terms of application, organ and tissue transplantation, you can use HLA for diagnosis also. In terms of the specific alleles, they have specific corresponding associated diseases. We go into that. And importantly, what is graft versus host disease, we will see in the next class, because it's one of the thing mentions about the transplantation barriers. Though you have a match, something may happen of post-transplantation. That what will happen, how it suits that, we will see in the next class. Coming on to serological testing, pros and cons. It's okay, it's minimal but fundamental, but there is a few other methods of advanced methods are there. It seems like we have less than 1 minute. I may not cover. Can I go ahead with sending one more link in 10 minutes? We can patch it because we can finish this, and Saturday we can go with. Okay, in 10 minutes or 15 minutes, I will send another Zoom link. 10-15 slides are there. We can finish this topic today. Okay, I will get back to you in 15 minutes in Zoom again. Thank you. See you soon.
Yes, sir. Thank you.
Okay, students, continuation of lecture 10. We have spoken about different HLA typing methods and in vitro methods to how to identify HLA match. And we briefly spoke about any barriers, transplantation barriers that may arise in terms of graft versus host reaction. Continuation of the HLA typing, because methods used for tissue testing, it's one of the curricular concept. Importantly, you go with whatever we discussed, that serological. You go with tissue typing in terms of mixed lymphocyte for matching T cell as such whole. And serologically, you go with individual HLA type or polymorphism you want to test. But as suggested, pros and cons in terms of serological testing, easy method, but requires large volume of blood. It takes time in terms of 3 hours of given the antibodies you have, and it also requires viable white blood cells that may not happen sometimes in the donor and recipient. It needs good anti-serum molecules in terms of antibodies. Where do you isolate? In terms of the quality of the antibodies. And it's really difficult sometimes to get the antibodies in terms of the cost effectiveness. But in terms of the match is not there, rarer antigens, you may not find a proper antibody, or you cannot raise the antibody in the anti-sera. In that case, do you mean that you cannot go ahead with transplantation for the recipient? You leave him just like that, I have no antibody for you, I couldn't test it in vitro, I'm sorry, you cannot say that. So to do that, what are the advances in molecular biology? You can go ahead, you can use specific genotyping methods. It's called a DNA genotyping technology. What you use with based on this, that you go specifically directly into, you identify specific alleles of the recipient, of the donor, based on molecular biology tools and a technique called genotyping. How do you do that? You know that we already seen the gene chromosome location. You know that specific genes where they are positioned. By doing so, by knowing so, you can design primers, just like a simple PCR test. You define primers for specific polymorphism, specific genes of HLA, class 1, class 2, and class 3. Based on that, you use PCR to detect the HLA genes in the donor, in the recipient. And if there is polymorphisms, you go with a test called restriction fragment length polymorphism. By separating the genes into small fragments, you look in the fragments which polymorphisms are there in which fragment. Either sometimes immediately you want to know the RFLP for the recipient, immediately go for RFLP for the donor. Don't need to test individually one by one gene. You can do that. And variable number tandem repeat, and also short tandem repeat, and also karyosome DNA sequence, next generation sequencing. This we don't go in deep. It's not required per se for the curricular aspects, but we will talk about one of the fundamental concepts of PCR labeling of HLA polymorphism or HLP genes. We talk how it works. Then we conclude with HLA polymorphism in the last class, a little bit more in depth, and then conclude the class for today. So we use polymerase chain reaction to identify sequence-specific primers. Through sequence-specific primers, and you develop a probes. Using that probes, you can go with the polymerase chain reaction. You can identify specific alleles or polymorphism of HLAs. But again, it is resources in terms of money, time consuming. Again, you go for one more advanced technique called RT-PCR. That is also better in terms of accuracy and quickness, but it's also in terms of the money is involved. If a patient is poor, whether they will afford or not, you have to look into that. You go with a molecular biology by PCR. How do you do that? Specifically, I'm looking for HLA typing by PCR SSOP. I'm looking for a gene in terms of DRB1. This is in the class 2 molecule of MHCs, and the DR and B1 is another molecule there. So I know the location in the chromosome, I know the sequence, I designed a primer. I can go with the traditional PCR reaction in the PCR machine, or I can take this sequence as a probe. I can put it into the glass slide, and then I can put specific primers or probes, whatever I designed. My probe will have a chemiluminescent signal. They will have a fluorophores, and then when there is a match on the glass plate, then the fluorophore excites, then it gives a positive signal. Then you go in recognizing the signal into the machine of chemiluminescence, you make a positive hit. Like that you go, and if you don't get a hit, then it is a negative signal. Then you might have to go with another probe, probe 1, probe 2. If you take a polymorphism DRBB1, there may be 140 polymorphism. Then you go with the 140 probes that you go by individually, but sequencing technology methods are highly advanced now. You go with multiplex mechanism. You go with giving all 150 or 800 at a time. The computer gives you that out of the 800, the donor or recipient, how many are matched. They can give immediately, shortest possible time, but the money is involved. But if you take a population also, the machines are so equipped now, you can put tons and tons of samples at a time, and they can give you the results in 2 hours. For example, if you give some 1,000 samples all over the world, just the machine runs it, all the probes multiplexing, it gives the positive hits in 1 hour, just like that. And it gives that of this world, whatever matched and where are the donor present, where are the recipient present in the database in the world. You can call the policies and database centers. You can approach and get the organ, whatever you want, in the shortest possible time. But money is involved again. So the procedure PCR SSOP relies on the locus specific amplification of the genomic DNA. If you are looking for a specific gene portion of the MHCs, specific alleles, so you go into single nucleotides also. The polymorphism or haplotypes, you go into the alleles also. The amplified DNA is mobilized to solid support. Then probes are developed. Probes are added in a direct hybridization. The probes are attached with the fluorochromes, and that will elicit a positive signal. A chemical signal can be detected. And as suggested, the resolution level is important. If you don't have enough probes, you may miss in the reaction. You go with usually 50 to 100 probes and then hybridize. But if a rare antigen, a rare haplotype, you might have to go with another synthetic method to develop the probe and go ahead testing it. That is PCR SSOP. Hopefully, you go with, if given a 4-mark question, given a chance of asking for tissue typing methods, you go with serological method. We spoke about testing the complement positive or not in vitro, getting the tryptophan signal. You go in that method. That is one more method of serological and HLA typing. Before that, you have to do blood typing test, briefly mention few lines, then go with advanced molecular biology technique. Given 2 marks, you write short. Given 4 marks, you write in broader way, if I presume. Both are covered in terms of curriculum tissue typing methods for organ and tissue transplantation. And we will quickly recap now a little bit more on HLA polymorphism. In the class of 9, I presume some of this terminology we went a bit fast at the end of the class. We will recap it a little bit slow so that you can understand what are the variability in terms of the genes, in terms of the chromosome, in terms of structure of the beta chains, alpha and beta chains, how polymorphisms can arise within the chain. So we have discussed in last class, there is polymorphism exists within species. There are multiple alleles for each gene, and it differs from species to species, human to human. And each variant binds different peptides controlling different antigen presentation to the T cells. So the peptides are nothing but the foreign molecules. The MHCs bind to the peptide. Each MHC will bind to each different peptide, and the combination as suggested MHC restriction. There will be a unique ternary complex, unique match for each of those. How they do that, we already discussed in last class. This is the class, this is the isotypes, this is the allotypes. You go with a complex that comes with either polymorphic or monomorphic. And how the variability comes up in terms of going to fundamentals. Each MHC allele can differ by 20 amino acids coming from what the alpha or beta helix is made of. Specifically, this is the cleft that helps in peptide binding for an antigen. So you can generate different variable of peptide binding by combination of alleles that will have different amino acids. They may differ, each allele will differ by approximately 20 amino acid change. That makes each allele quite unique and distinct. Most of these differences are located in this cleft that helps in binding to specific antigen. That what was the peptide binding properties. Zoom in terms of, again, what are these we spoke in the last class. Let's take a little bit 1 or 2 minutes here. We are looking at the class 1, class 2 here. What are the MHC molecule in terms of the polymorphism, in terms of the type we are looking for. In class 1, there are 2 types, HLA-A, HLA-B. Again, different polymorphism, it's 02 and 01 subtype. It's a 27 and 05 subtype based on the match here. And this specific MHC molecule always associated with specific peptide coming from foreign antigen. That is that second bound peptide, HIV reverse transcriptase. It is relevant to HIV, human immunodeficiency syndrome disease. And this specific antigen from HIV binds to specific class 1 molecule MHC here only. Then this is influenza. This comes and binds to specific HLA in class 1 again. Specific different class of HLA-A, and type is B here. And this is association. It is unique. And how it is happens? Specifically, it happens through peptide binding motif. It is relevant to the peptide binding motif here. Residues you are looking in terms of the MHC molecule. In terms of this is a bound peptide, and the down you are looking for HIV reverse transcriptase. They have a specific match given the residues of the position. N to C, 9 residues are here. Usually the class 1 HLAs will have a kind of specificity in terms of position residue 2 and 9 that called monomers. And they will have a unique combination, bound peptide from the foreign antigen, foreign molecule, virus or bacteria, some other molecule, and the peptide binding motif, they will have this match here. The same consensus for in terms of class 1 monomers, again different molecule, different foreign antigen, but a consistency of 2 and 9 location, you will have a variability here that associates. Coming on to that, class 2 molecules, as suggested, class 2 are always CD4 associated, and they are always associated with antigen presenting cells. So you are looking for specific kind of your own immune cells. For example, here antibody, light chain of IgK, and also transferrin receptor. This is one of the T cell pathway. These two are self molecules, and that's why class 2 associates with them. They have a variable location. The self peptides vary accordingly. They don't have some monomer consensus sequences. So based on this suitability of molecules matching with the bound peptides, there is some consensus in terms of the peptide binding domain. So that makes it that unique in terms. At the same time, it makes variable in terms of that a peptide binding motif, a combination of anchor residues. These are important. They are the one can generate variability. If anchor residue from this class varies, it may differ to in terms of the bound peptide. The class also changes here, so resulting in susceptibility or resistant to a specific disease that affect binding. Thus, bringing on to the conclusion that MHC molecule in terms the polymorphisms, why they are important. At least 3 properties of MHC molecules are affected by the polymorphism. Because of this polymorphism, they can bind to a range of peptides. They can change the conformation of the bound peptide accordingly to the polymorphism. The alpha and beta chain or 2 alpha chains and the peptide motif will change the conformation. They bind to a specific antigen. An interaction of these MHC molecules directly with the T cell receptor is also influenced by this polymorphism in the repertoire here. So that basically tells in terms of MHC restriction, what we discussed in the first half of the class, a unique ternary complex, it has a unique specificity. It may not bind to a different antigen, though both complexes are present, antigen is different. Though antigen and T cell receptor are common, but HLA type has been changed, MHC molecule, it will not fit into that. This is an important concept. So basically that concludes the class for now. But coming on to the COVID-19 situation, what we are looking up, there is immunology is important, right? Immunology of individual haplotypes of a population, haplotypes of a regional location is important. How do you are susceptible to COVID-19, the virus, or you're not susceptible? So there is a kind of evolutionary trend that usually we observe. Given a population, given some 2 centuries, we have got a lot of pandemics, right? Humans have this encountered a lot of pandemics. This slide is for general interest. I was take few minutes, maybe 1 minute, and quickly finish it up. What we are saying here in terms of the COVID-19, in terms of how the immune system can help you to either susceptible or resistant to a given pandemic. Basically, you're saying a concept of evolutionary traits, the fitness of an evolutionary trait given a population. Species survive, individuals die. So pandemics come and go. A lot of pandemics and epidemics has come. But as a species of humans, we are not eradicated completely. We are still surviving. We are still existing. Few people died, individual population died. But as a species, we survived. How did we survive? What it made us to survive? It's nothing but we need to thank our immune system. We need to thank specifically our MHC polymorphisms, haplotypes that are quite strong and robust of given a population, given a polymorphism of a species. Some polymorphisms were not fit, they died, they couldn't be able to protect you from the foreign invading antigens, but then majority of them are robust. They are preserved given an evolutionary test, evolutionary fitness. They survived and they sustained. What does it say here is that given a pool of MHC haplotypes here, given an epidemic disease, for example, smallpox, diphtheria, cholera, influenza, they have come up and some population of individuals died, basically saying that some of the haplotypes are not fit to the given condition. They are eliminated during evolution. Like that, there's some survived, there is again harmony will be there. There is again weak selection because some are died. And later, again, you grow in the population, you come up, but again a new epidemic comes, new pandemic comes, for example, COVID-19 or HIV or AIDS or MERS, SARS, whatever flu we encountered. Again, some survives, again go into second selection, you go into that, a reduction of 75%, because in this situation, we are tested very keenly by COVID-19 that I'm going to win over the human population. It is saying that. So a kind of 75 reduction may happen given a scenario of 6 months. Haplotypes may not survive, but you consistently look in specific populations here, they survive. The haplotypes, they still survive. They fight against the pathogen. They say that you cannot win over the war. I am here to help the human population, human species. I sustain, I am kind of resistant to anything, and a kind of small population, they make us survive and they make us the fit. That is basically we should thank our immune system. We should thank our immune MHC haplotypes. That comes to a conclusion of the class. We will take further, I think probably the videos I can put in the links. Some Flash player you need to, Adobe Flash player you need to activate to see some of the videos. You go into that. After that, we will resume a class on Monday. We go into grafting and then you go into graft versus host disease or reactions. Okay, I will see you on Monday.